In the name of Almighty God, I am Mahsa Sankoshi, and today I want to talk about particle trajectories. Refers to the curves traced out in space by a classical point particle as it moves, described by its position vector over time. In fluid mechanics, these trajectories can also be referred to as material pathlines, representing the motion of fluid particles within a continuum. In static mixers, also called motionless or inline mixers, a fluid is pumped through a pipe containing stationary blades. This mixing technique is particularly well suited for laminar flow mixing because it generates only a small pressure losses in this flow regime. This example studies the flow in a twisted blade static mixer. It evaluates the mixing performance by calculating the trajectory of suspended particles through the mixer. The model uses the laminar flow and particle tracing for fluid flow interfaces. Particle trajectories are the paths traced by moving particles analyzed in fields like physics, fluid mechanics, and cell biology to understand motion, interactions, and dynamics. In classic physics, trajectories are the spatial curves described by a particle position vector over time, often determined by external forces such as electric or magnetic fields. In experimental contexts, trajectories are observed as sequences of discrete data points acquired from images, providing a time series of a particle's location. And what are the key aspects of particle trajectories? The first one is definition. The curve followed by a particle as it moves through space. The next one is data collection. In experimental physics and biology, single particle tracking or SPT captures time series data of a particle's coordinates or x, y, z. The next concept is influencing factors. External factors like electric and magnetic fields, particle-particle interactions, and the properties of the surrounding medium influence the trajectory. The next concept is analysis. Statistical methods and modeling are used to analyze trajectories and extract information about the underlying dynamics, such as the diffusion coefficient in random motion. What are the applications of trajectories? Physics. Understanding the motion of charged particles in fields such as the circular path of a proton in a magnetic field. The next application is fluid mechanics, analyzing fluid particle motion pathlines in a continuum. The next application is cell biology, observing and analyzing the motion of individual molecules or cellular structures using single particle tracking. The next application is in engineering, designing and optimizing particle-based devices like electron microscopes by predicting particle passes. And how trajectories are determined. The first one is theoretical calculations using equations of motion such as those derived from Newton's second law to predict a particle's path under specific forces. The next one is simulations. A specialized software simulates particle behavior under various conditions and within complex geometries to analyze and predict trajectories, such as Comsol software. The next one is experimental observation. High-speed video cameras and other microscopy techniques are used to capture the movement of individual particles, creating a series of position points that form the trajectory. The next one is single particle trajectories, or SPTs, consist of a collection of successive discrete points casual in time. These trajectories are acquired from images in experimental data. In the context of cell biology, the trajectories are obtained by the transient activation by a laser of a small dyes attached to moving molecules. Molecules can now be visualized based on recent super-resolution microscopy, which allow routine collections of thousands of short and long trajectories. These trajectories explore part of a cell either on the membranes or in three dimensions, and their passes are critically influenced by the local crowded organization and molecular interaction inside the cell. As emphasized in various cell types such as neuronal cells and astrocytes, immune cells, and many others. And about SPTs allow observing moving molecules inside cells to collect statistics. SPT allowed observing moving particles. These trajectories are used to investigate cytoplasma or membrane organization, but also the cell nuclear dynamics, remodels, dynamic or mRNA production. Due to the constant improvement of the instrumentation, the spatial resolution is continuously decreasing, reaching now values of approximately 20 nanometer, while the acquisition time step is usually in the range of 10 to 50 milliseconds to capture short events occurring and happening in live tissues. A variant of super-resolution microscopy called SPT-PAL is used to detect the local and dynamically changing organization of molecules in cells or events of DNA binding by transcription factors in mammalian nucleus. Super-resolution image acquisition and particle tracking are crucial to guarantee a high-quality data. The next concept is assembling points into a trajectory based on tracking algorithms. Once points are acquired, the next step is to reconstruct the trajectory. This step is noun tracking algorithms to connect the acquired points. Tracking algorithms are based on a physical model of trajectories perturbed by an additive random noise. The next concept is extract physical parameters from redundant SPTs. The redundancy of many short SPTs is a key feature to extract biophysical information parameters from empirical data at a molecular level. In contrast, long isolated trajectories have been used to extract information along trajectories, destroying the natural spatial heterogeneity associated to the various positions. The main statistical tool is to compute the mean square displacement or MST or second order statistical moment. The next concept about the particle trajectory. The life history of a particle is built up from a knowledge of its trajectory through the particular system of interest. Consider the path of a particle as it travels through some homogeneous medium. Since the typical particle scatters frequently, the path will zigzag rather than in the manner indicated as you see in this simulation. Here the particle originates with known direction and energy. It has a free flight until it has a collision with an atom of the medium. This collision could result in the absorption of the particle and the immediate termination of its history, but we shall assume a scattering interaction happens and the particle continues with a new direction and a change of energy or a wavelength. This change of energy and direction is a statistical process, that is, there is not a unique energy and direction after a scattering. Rather, there is a probability distribution for each of these variables. After the first scattering, the same particle makes another free flight and experiences another collision, and so on. And The next concept about particle trajectories. The CFD or computational fluid dynamic model computes the particle trajectory using a Lagrangian formulation which includes the inertia, hydrodynamic drag, and the force of gravity. The trajectories of particles determined will only be possible in freeboard as the particle concentration is quite less compared to gas, whereas in the dense bed with DPM Lagrangian approach and with present computational power, it is not possible to track the particle as concentration of particles in dense bed is quite high as compared to gas. The particle trajectory will be depending on size, density, and moisture content in the particle. The history of each single particle can be tracked both qualitatively and quantitatively. The trajectories of particles can be plotted by selecting the desired number of streams. Besides, the DPM report gives information about the average residence time of the fuel particles in the freeboard and the combustion efficiency. Both greatly differ from case to case. As these particles are denser and contain more moisture, these will vanish in bed instead in freeboard. The ash particles, however, will be ejected out of furnace. Now you can see my simulation here that I have used particle tracing for fluid flow, physical module, and also laminar fluid flow. And you can see that I have selected two study, stationary and time dependent. And after that, also you can see that which kind of material I have used here. It's a user defined material with a specific density and dynamic viscosity. And now you can see the velocity of this particle, and the unit is meter per second, and the pressure, particle trajectories. You can see the direction of particle here, and also with this FPT. There is a maps of these trajectories and phase portrait. Thank you so much for watching my video until the end. You can also download this Comsol simulation from my github.com master script.